we have my talk. So just to respond to the last question, I'm talking about personalized medicine. The first line is, here. Green button, okay. Okay. The basic problem, one basic problem in medicine has been for a long time that we are all different. Every patient has a different genome. Tumors arise in different somatic cells of patients with different genomes. Tumors are heterogeneous. So no tumor has ever been seen before. No tumor will ever be seen again. In fact, no patient has ever been treated before. So we have a situation in which that is taken care of properly in some areas of medicine where we have true personalization. If you go to a hospital with a complex break in your arm, the doctor can take the imaging tools to decide exactly what the situation in you compared to the next patient is, and he is able, based on his experience and the structure of the human brain, to anticipate which therapy will be best for the patient. So that's quite different to what happens to you if you get a drug. Drugs affect, as medical entities, complex molecular networks which differ from patient to patient, from tissue to tissue, and we neither have had the information on those complex networks, nor has the human brain the capability to predict the action of complex drugs with complex effects on such complex networks. So basically what happens to you if you get a drug is the equivalent of going to a hospital and getting the right arm in a cast, even if you have broken the left arm, because in a clinical trial there were more patients with a broken right arm, therefore cast on the right arm has become this first-line treatment. This has all sorts of consequences, all of which are negative. That's success rates, the response rates of the 10 most highest grossing drugs in the US. And the blue figures symbolize the fraction of patients responding to those drugs. The red ones, those not responding. So you see the overwhelming majority of the patients who get those drugs do not respond to the drugs. But they can have side effects, adverse drug effects. In Europe, kill more people than colon cancer, close to 200,000 per year, and the annual economic costs in the US are estimated to be between $330 and $1,130 billion. All that contributes to the enormous healthcare costs everywhere in the world. Europe spends per day 4.5 billion euros on healthcare, the US roughly 10 billion, and not only that, but the costs increase by 2% faster than the gross national product, which is clearly already unsustainable in a stable society, in an aging society. It will have disastrous consequences. We have now, through the sequencing, enormous progress in sequencing technologies, the capability to analyze the genome, where we had before the capability to sequence 96 sequences in parallel in the machines we used in the Human Genome Project. We can now generate billions of sequences on a single machine. We can sequence with the newest generation of machines something like 60 genomes in a single run lasting 2 days. So the costs have dramatically decreased much faster than the costs of computing. Now, there are two levels on which this is used. For example, in cancer care, we are on one hand using some molecular characterization by sequencing, ranging from individual variants to what we do, a very deep analysis, low coverage whole genome, deep exome of both tumor and patient, and a deep transcriptome analysis. So we generate typically both in our spin-out and in our research projects hundreds of times more information than, for example, companies which have which are very well known in this area. So, and that illustrates how you can identify many therapeutically important variants which you will never see in the low coverage analysis. In particular, if you do not analyze the patient genome, you don't know if the variants you see are really somatic, have a reason during the tumor formation, or pre-exist in the patient genome, which gives them a different functional importance. And particularly, without deep transcriptome, you miss epigenetic effects which can just as easily silence, for example, recessive oncogenes as a mutation, which is the only thing you would see in a mutation screen using the genome. Now, this is a very powerful tool to stratify patients. We can identify actionable variants. We can identify overexpressed pathways, but it doesn't give us proper personalization, personal medicine. For that, we need to apply a tool which is actually standard in all other areas of our existence. The fact that in complex systems, we cannot avoid making mistakes. We can only make sure that we make mistakes without consequences to people, without costs, without losing time. If you build a skyscraper, you build it first in the computer and test it against all possible challenges before you actually set it up in reality. Same thing for cars, planes, or even electric toothbrushes, except for medicine and clinical trials, which still experiment basically in in the therapy and in running clinical trials, in contrast to, for example, virtual crash tests. The reason for that is simply that the technology hasn't been there to initialize the models. You need a huge amount of information to be able to take a mechanistic model and make any useful predictions. You can't even make useful predictions if you have only in a weather model the temperature in front of your house. And that's exactly the situation we have been in medicine till a short time ago. Now, due to the enormous progress in DNA sequencing, we are now in a situation to initialize complex models of tumor and of the entire patient, treat those models with drugs, and observe both effects and side effects. And this is a case in which we have been early on very successful. For example, we have had in a research project a patient with metastatic melanoma who had to stop the standard treatment, and therefore the doctors tested the drug which looked best in the modeling. Essentially, the blue squares indicate a patient which is the rows, and a drug combination, drug as a columns, which had the effect of lowering the MYC concentration in the tumor as a surrogate marker of cell division. And so that patient had shrinkage of the metastasis and was stable for more than a year. Now, the counterpart to that is actually the drug development, which works by the same type of concepts. Drugs are enormously expensive because of the very high wastage. If you divide the costs of the different drug development programs by the drugs which actually get approved, you end up with numbers of between 4 and 12 billion euros per drug, buying in many cases only fairly moderate medical benefits, short extension of lifetime at relatively high cost. The reason for that, I would argue, without having the time for it, is that basically all drugs which are anywhere in development might very well help patients. We just have to find out through the modeling which patients, and ideally as early as possible run clinical trials immediately after synthesis of the compound, rather than a real clinical trial costing $500 million, which then fails in the late stage of clinical development. So those techniques are obviously still expensive. Anything new tends to be expensive, but partly because we can't use the most powerful machines, but also we have some, but also we have to automate the process to make them less manually work intensive. And in other areas, for example, in the development of solar cell industry, the government therefore has initialized the process with hundreds of billions of taxpayers' money. I wish we would be considered as cancer patients as important as solar cells or electric cars, which are subsidized by 4,000 euros in Germany. All models are wrong, some are useful. We try to generate useful models, and one of the tools which we are working on is, as Angela has mentioned, a flagship project which might provide 1 billion euros over 10 years, roughly the amount of money we probably waste in healthcare within roughly 12 hours by prescribing the wrong drugs. This might hopefully generate models which can accompany you through life and which can die many times over or during the course of your life, protecting you against any problems due to the non-personalization of everything you're exposed to. So our key people have contributed to the work, and I hope you will all be convinced that maybe this is worth thinking about. Thanks.
So the question I had last time also fits this time. So the health system is under tremendous cost pressure. So are there, in this, you mentioned also cost, and I'm wondering in which kind of context or how the cost is being evaluated, because on one hand, one size fits all seems to be very cost efficient. On the other hand, you add a lot of analytical tools also to this. So how is this in this community being discussed?
It's a very cheap way to kill cancer patients, admittedly. Simply, the adverse drug reactions cost a significant fraction of the total healthcare expenditure. If you count in the downstream consequences, additional medical treatment, loss of working time, I'm sure that the 4.5 billion we spend per day can be cut by at least 1/3 till 1/2 by a true personalization of therapy. I would argue that development of drugs could be, the cost could be reduced by 2 to 3 orders of magnitude without being an expert, but simply because we could get many more drugs to the market in a much shorter time at much lower costs, eliminating the drugs which do not work early on, replacing and improving preclinical models, because you essentially model the mouse just as much as the human, and then you can translate your conclusions from the mouse much better into the human system and so on. But I'm happy to discuss that with anybody. I'm convinced this individualization, use of the full power of computer systems and analytical techniques improving by 3 orders of magnitude every 10 years is the only way to keep up a sustainable healthcare system.
Thank you, Hans. Thank you very much. I'm sure that Ansgar, you will pick up, I think, one of